Welcome back. This video is the continuation of part 1. In the previous video, I have described the properties of light and how light interacts with the object. Magnification power, resolution and numerical aperture are also described, and I have also mentioned that microscope is an optical instrument that is used to study small objects, and it consists of one or more than one lenses. And depending on that, we can categorize microscope into two types. In simple microscope, a single spherical lens is used for magnification, and that's why magnification is fixed, and generally it is used for small eukaryotic cell observation. The size of the simple microscope is small and the design is also simple. It is not used in the laboratory nowadays. But in compound microscope, more than one lenses are used for magnification, and here magnification can be increased by changing lens combination. It is used for both prokaryotes as well as eukaryotes. It is larger and complex in design than simple microscope. It is used for routine microscopic study. From those differences, we can understand although simple microscope is cheaper than compound microscope, but due to some limitations, it is replaced with compound microscope. Here I am going to discuss compound microscope only. Depending the source of illumination, microscope are further divided into two types. In light microscope, visible light is used for illumination and glass lenses are used for magnification. In electron microscope, beam of electrons are used, and instead of glass lenses, electromagnetic lenses are used. Lots of other differences are there between light microscope and electron microscope, but it will be discussed later. Now in this video, we will talk about compound light microscope only. Compound light microscope are again divided into many types depending on how contrast is generated between objects and background. Now what is background? If you want to observe red blood cell, then red blood cell is your object of interest. But before observation, you have to make a uniform layer of blood onto a glass slide in order to fix it on a transparent and solid support. The layer of blood should be so thin that can't able to block the passage of light through it. Here glass slide is our background or field on which you are not interested. Now let's see the classification. In most of the laboratories, these four types of optical microscope are used. Under this type of microscope, object looks darker than the background. Under this microscope, object looks brighter than the background. And here, contrast is generated by changing the phase of the light into change in the intensity of the light. A fluorescent dye is used only which emits the light, and the background is completely dark in case of fluorescent microscope. The detail will be discussed later one by one. Apart from this, some special purpose microscope are also there. They are stereo microscope, inverted microscope, metallurgical microscope, polarizing microscope, confocal microscope, scanning probe microscope, and super resolution microscope. All microscope have some special features and designed to use for some special purpose only. The most commonly used compound light microscope is bright field microscope. So let's start with bright field microscope. First, let's have a closer look to each and every parts of the microscope. This is the power on off switch. You can switch on or off the source of illumination using this. A wheel is also attached here to control the light intensity. The light will pass through this lens called condenser. The function of the condenser lens is to focus the light onto the object. An aperture is also there in this position to narrowing the beam of light. The utility of the aperture will be discussed later. This area is called stage. To keep the slide steady, you can fix the slide onto the stage using those clips called slide holder. If you want to change the position or want to scan the whole area, then you can move the slide using the stage controller. These are called objective lenses. They are the heart of a compound microscope. In modern compound microscope, nearly 6 lenses are added, one behind another. That's why they are called compound microscope. You can interchange the objective lens depending on your requirements by rotating this nose piece. Generally, 4x, 10x, 40x and 100x objective lenses are commonly found in the compound microscope for routine use. 4x objective lens is called scanning because it is used for scanning the whole slide to check where the object is. 10x is called low power. It is also used for general view without much detail. 40x is called high power because it gives a much detailed structure of the specimen, and 100x is called oil immersion. It gives you the most powerful magnification. A special oil is used to fill the gap between slide and lens. The name of the oils which are commonly used are cedarwood oil, mineral oil and balsam. You can find more detailed description and characteristics of each objective lenses in this table. You can see when the numerical aperture of the lens is increased above 1, oil is used and the working distance also decreased. Apart from this, specialty objective lenses are also there. Two wheels are there. First one is called coarse adjustment for moving the objective lens up and down for focusing the object. After adjusting the coarse focus, you have to fine tune the image by rotating this fine adjustment to get a clear picture. This area is called arm for holding or lifting the microscope. Again, two lenses are here in this position. They are called eyepiece or ocular lens. You have to see through these lenses. Generally, the magnification power of the ocular lens is 10x, but sometimes 15x are also available and it is fixed. If you are using 40x objective lens, then the total magnification will be 400x. If only one ocular lens is present, it is called monocular microscope, and if two ocular lenses are there, it is called binocular microscope. Unlike monocular microscope, in binocular microscope, you can further adjust the focus and get a 3D view of the image. Now, how to operate a compound microscope? Your first task is to switch on the main power, switch on the light source, place the glass slide onto the stage and rotate the coarse focus first. Fine tune by help of the fine focus, observe through the ocular lenses. Now, what is the principle of compound bright field microscope? Light rays are focused onto the object by help of a condenser lens. Depending on the nature of the object, light rays either absorbed or refracted or transmitted. The background do not absorb any light. The light that comes from object and background then entered into the objective lens. It actually enlarges the object. Depends on the magnification power of the objective lens. Light then pass through the ocular lens, which further magnify the object and reach to the observer eye. The observer will see a magnified virtual image in this plane, but the image is inverted. Here, magnification achieved in two steps. First, the objective lens produce an enlarged image of the object in this plane called real image plane. This real image is further magnified by the ocular lens or eyepiece to produce the virtual image. The function of the ocular lens is to spread the light rays so that they appear to come from a large inverted image from this plane. Because light rays are not actually passed through this location, that's why this image is called a virtual image. The object looks slightly darker than the background, depending on the diffraction, absorption and transmission property of the object. In order to increase the contrast between object and background, or to distinguish some special structure from the rest of the cell, like endospore of a bacteria, chemical stains are used. After staining, some part of the cell looks colored and some are not. Bright field microscope is a multi-purpose, regular instrument that can be used routinely for both live, unstained material and preserved stained material. Because of some advantages, bright field microscope are generally used in the lab. For specimen preparation and observation, expert technicians are not required. Can be used for both stained and unstained specimen. Color of the specimen is not altered. If camera is attached, a good quality picture can be captured. Stains are used for staining and that are cheap and easily available in the market, and it is cost effective. Although bright field microscope is used routinely in the laboratory, but it has some disadvantages, like highest magnification is 1000x. It is not a very good option for observing living small organism. Contrast is low, so in order to increase the contrast, stains are used. Proper knowledge for staining is required, and sometimes multiple steps are there and it is rigorous. Staining and fixing may distort the original shape of the object. Bright light used for illumination and can produce heat that will damage the specimen or kill living microorganism. Working for a longer duration may cause damage of the eye. Now let's check some commonly used terms related to microscope. First, what is projection distance of a microscope? It is the distance between the virtual image and the eye point. Here in this picture, this is the projection distance. And next, what is depth of a focus? It is known as the depth of sharpness or penetration. The depth of focus of an objective depends upon the numerical aperture and the magnification, and it is inversely proportional to both. So that's all for now. If you like this video, press the like button and don't forget to subscribe my channel and write your comment in the comment section and share the video with your friends, and thanks for watching.